Okay, thank you very much, and sorry again for the technical difficulties. I'll try to move at a quicker pace to get through in a timely fashion. First, I just want to thank Gene's group for allowing me to do this project, and the team that helped me with the project is on the right here. I'll go through these folks one at a time as we get to the work they did. And just by background, by way of motivating the study itself, we wanted to increase the detection rate for RNA-protein interactions. So RNA molecules, which are central to gene expression programs, are coded throughout their lifetimes by RNA-binding proteins, or RBPs, as I'll call them. And RBP interactions with RNAs control all the various processing steps that are diagrammed here. And each one of these steps is disrupted in human disease. And in fact, more RNA-binding proteins are associated with human disease, with genetic human disease, than are transcription factors. So they're an important class of proteins to study for developing therapeutics and for understanding human disease. Of note, neurological diseases are especially sensitive to aberrant RBP function. This has to do with the fact that they utilize RBPs to control gene expression in very regulated ways, with these phase-separated condensates that control localized translation and also RNA processing in the nucleus. And so highlighted here are all the different RBPs that are mutated in neurodegenerative disorders. And single-cell RNA-seq and multi-omics approaches have been powerful for exploring these neurological disease mechanisms because they can give you the cell type specific gene expression landscapes. Neurons are very heterogeneous cell types, and these sorts of approaches allow you to get at the cell type specific gene expression that's disrupted in these diseases. It would be nice, given that RBPs are often mutated in these diseases, to understand at a single cell resolution RNA binding profiles and also the actual protein output or translatomes of these neurological cell types in a cell type specific manner. Another human disease with aberrant RNA or RBP malfunction is cancer. And so again, at each step in the mRNA life cycle, these RNA processing events driven by RBPs are disrupted. And importantly, I'll highlight translational control driven by RBP remodeling of the translatome is important for driving cancer cell plasticity and drug resistance. And then again, single cell approaches have been powerful for defining the heterogeneous tumor and tumor microenvironment, here shown for breast cancer. And so this translatome remodeling actually gives rise to this drug-resistant plasticity by giving rise to these cancer stem cell-like populations. So again, it would be really nice if we could probe the RNA binding landscape and the translational output at single cell resolution for these types of disease models. So this is basically the multi-omics single cell toolbox that exists to date. So there are many ways to probe genomic and epigenomic profiles or signatures from single cells and to couple that to single cell RNA-seq, and more recently, proteome evaluation at low throughput for surface proteins is also available for single cell approaches. So we wanted to add to this toolbox single cell resolution measurement of translatomes and RNA binding landscapes. So these are the gold standards for measuring RNA binding protein interaction with RNA and also ribosome association with RNA, CLIP and RiboSeq. And I'll walk in more detail through the overview of the general CLIP workflow. So pre-processing, you cross-link RBPs to target RNAs. Following cell lysis, you fragment RNA and digest with RNases. And then you isolate the protected fragments or the cross-linked fragments with immunopurification and page gel purification of the RNAs bound by the target RBPs. And after digestion, you ligate adapters. Reverse transcription and PCR amplification will give you read densities that overlap with cross-link events, which gives you a sort of a read peak density above binding sites. So there are some limitations to the CLIP approach shown here. So you can have where there's cooperative binding between two RBPs or complex binding, some false positives and false negatives that arise from cross-linking of complexes as opposed to individual RBPs. There's also occlusion when there's antagonistic binding that will give you false negatives, and then also weak binding or transient binding will give you also false negatives. Strong binding probably gives rise to most of the real signal, and then there's stochastic expression. So in individual cells, the target or the RBP of interest might be expressed at different levels, giving rise to false positives and false negatives. And then fragmentation of the target RNAs limits the ability to determine isoform level information about what target is bound. And then the performance of the antibody can also give rise to false positives if it non-specifically binds an off-target RBP, or false negatives if there's low efficiency of antibody binding. And then RT, or reverse transcription, and then PCR amplification also give rise to artifacts. Until recently, PCR duplicates were a main source of false positives or artifactual signal in CLIP experiments. And our lab recently was able to get around PCR duplication artifacts by increasing the efficiency of ligation of adapters. And so Eric Van Nostrand pioneered a new CLIP protocol, and then in conjunction with other labs in the ENCODE project, was able to increase the throughput for CLIP evaluation of binding sites and other functional assays for hundreds of RNA binding proteins. And so this was a massive effort that gave us lots of insight into the functions for many RBPs. So we wondered if we could develop more scalable methods to identify RBP targets, and then a method that would potentially fill in the blind spots, so the isoform aware and single cell resolution information for RNA binding profiles. So recently there have been developed RNA tagging approaches. So if you fuse an RNA editor or modifier to an RBP of interest and express it in cells, it will leave a calling card on the bound RNA where it has bound, and you can just through standard RNA sequencing without having to do immunopurification determine where the RBP has been. So this was first demonstrated in 2015 by the Wickens lab, where they in yeast used polyupolymerase to tag target target RNAs by fusing to an RBP of interest. And this approach gives you target level information, but not binding site level information, because it's appending the tag on the ends of the RNAs. Then after that, my Grasbach's group had developed in flies the TRIBE approach, which fuses an RBP to A to I editor, RNA editor, and this allows internal RNA tagging. And the TRIBE approach, so-called, worked for internal binding site resolution, but it was a little sparse in signal and low resolution compared to CLIP. So if you look at this example gene, you have several edits here that occur on this target gene, but they do not overlap with many of the CLIP peaks that are elsewhere on the target gene. And if you look at the average number of edits per gene, target gene, 1 or 2 edits per target gene is kind of the standard for the TRIBE approach. This is because the sparsity of signal is because ADAR relies on a double-stranded bulged substrate, which are infrequent in single-stranded mRNAs. And so to get around this, the Grasbach group has used a hyperactivated mutant of ADAR, which increases the editing rate from generally 1 to 2 sites per target gene to maybe 5 to 10 sites per target gene. So this is a so-called hypertribe update to the TRIBE approach. And so comparing a poly tagging and TRIBE approach to CLIP, what you get is antibody-free detection of these targets. What you lose for poly tagging is at least binding site resolution, and for TRIBE, signal on single-stranded RNAs. So to get around this, people have used a new editing enzyme modality. So APOBEC is a CDU editor that is more highly active than ADAR on single-stranded RNAs. And use for this module was demonstrated by Kate Meyer's group a couple years ago, where they fused a YTH domain. So this is a domain of RBPs that generally read this RNA modification M6A. And this fusion was able to detect M6A modifications in a very sensitive way in human cells in response to perturbations that increase the level of M6A across coding sequences. You can see that this signal is more well distributed than what you get for A to I editing with an ADAR enzyme. So we wanted to utilize this new module with increased signal to potentially do a few things. One would be to see if we could do full-length RBP binding site detection in cells using this module. And then if we could do ribosomal subunit fusions to detect ribosome association or translation from target genes. And then also to use this increased boosted signal to do isoform specific binding from long reads approaches, and also lower the input down to single cell resolution binding sites. So the strategy here was termed STAMP. And so as a control, we express the enzyme alone or fuse to an RBP of interest. CDU edits, again, give rise to these mutations, which can be detected by standard RNA sequencing. And here I'm just going to show you an example gene. Before I move forward with this data, I just want to make sure that I'm still online because I don't see chat in my screen. Just somebody say.
Yeah, you're still online.
Okay, great. Thank you.
And I'll just reiterate, if anybody wants to jump in and ask questions, feel free.
Okay. So this is an example of the approach and the signal that we get from it on a target gene for the well-characterized RBP RBFOX2 on this target gene APP, where you see that these clip peaks are in the 3' UTR and not in the size-matched input control. And when we express the ApoBec enzyme alone, we see some low-level mutation rate that occurs at increasing levels of expression. And then RBFOX2 fusion, when we increase expression, we see these mutations start to crop up. And then in gray, what we're just looking at is the RNA-seq coverage. And then when we use a computational workflow to quantify these edits, we see that the edits quite well track the clip peaks in density. And further, we can filter out using confidence level filtering of the edit sites to where only one site is surviving in the control, but many edits are surviving the cluster around, again, the clip read densities. This editing, this directed editing is very reproducible. So increasing levels of the transgene expression, we see very nice reproducibility genome-wide across replicates. It does require overexpression of the fusion. So there is some leaky expression from these stable cell line inductions. And then at low levels and at high levels, we see roughly about 1.5-fold increase in expression of the transgene compared to the endogenous RBFOX2. And then overexpression of these editing fusions actually does not have a significant influence on gene expression. So comparing to CLIP, we see that if we center around the known RBFOX2 motif genome-wide, we have density that centers around where these motifs are on binding sites, and it is in concordance with increased significance of CLIP signal. We get increased density around this motif for the edit clusters. And we can just throw the sequences around the edit clusters into Homer motif extraction software, and it very reproducibly and significantly enriches for the known RBFOX2 motif in all genic regions where the edits occur. So Eric Kaufman then developed a denoising computational pipeline to filter out what are considered to be background edits. And here this table is just showing you that we can go from roughly 400,000 edits for RBFOX2 down to 20,000 highly significant edits, and this results in like 5,000 edit clusters, or you can consider these sort of similar to what we call clip peaks from a clip experiment. And comparing to the control, where you remove basically all significant edits and edit clusters. So
Chris,
looking at, yes?
Chris?
Yep.
Can I just jump in real quick? Doesn't the fusion of the ApoBec1 together with the RBP, doesn't that influence the RBP binding?
Does it influence the binding profiles?
Yeah.
Yes, we do think that that is the case. So when we clip an ApoBec RBFOX2 fusion with a tagged antibody or HA antibody, it does overlap with a majority of the clip peaks from like an endogenous clip experiment, but there are increased 3' UTR clip peaks from that particular fusion. We think that has to do with the overexpression causing a more cytoplasmic localization, or potentially the fusion itself causing a more cytoplasmic localization. So it is important, I think, for each use case to evaluate how the fusion and the overexpression affects the subcellular localization and the binding profiles. And the way to do that is through immunofluorescence and through clip of the fusion itself. And once you've established what the differences are, you can use that information to sort of dial expression or adjust fusion orientation or linkers to sort of optimize towards a more endogenous binding profile.
Okay, thanks.
Yep. So looking at the fraction of clip peaks that are captured by these windows, we see that without filtering, we get roughly 50% of the known clip peaks are captured. And then we shuffle regions of the same size as these edit peaks. We see a much reduced capture of or overlap of these regions with these shuffled regions with these clip peaks. And this sort of ratio is maintained at higher confidence levels and with all filtering thresholds in place. And when we take the most highly significant editing clusters or edit peaks and compare them to clip, we see that more than half overlap known clip peaks or motifs, or clip peaks containing known motifs. And then of the 45 or so percent that do not directly overlap clip peaks, more than half of these actually are on clip targets in the vicinity of known clip peaks. So fairly good overlap between these highly significant edit clusters and known clip peaks. So yeah, while this isn't perfect, this is the highest resolution sort of antibody-free RNA tagging approach compared to clip so far. And we did this, we demonstrated that this works for several RBPs. So looking at SLBP, which does not bind polyadenylated RNAs, but binds these structured 3' UTR stem loops of histone genes. And we see very specific editing within the regions adjacent to these structured stem loops on histone genes. And then for TIA1, which has a known U-rich motif and binds 3' UTRs, we see overlap with CLIP that gives rise to this known U-rich motif for TIA1. So it works for multiple RBP backgrounds. So what fun things can we do with STAMP now that we've established that it works? What we wanted to try is long read evaluation of isoform specific binding so we can get alternative 3' UTRs or alternative polyadenylation events. And you can imagine that if an RBP is bound here between these two different isoforms, you would not be able to resolve this binding event with a CLIP experiment. But long reads might tell you that it's binding this long one as opposed to the short one. And RBFOX2 has been shown to influence alternative polyadenylation by binding 3' UTR regions more recently. So looking at RBFOX2 and long reads, comparing to CLIP, again on the APP gene, you see the background control, you see short reads enriching for edits around the CLIP peaks, and then direct long read using the Oxford nanopore system, we see that there is some background editing from the control, but then you enrich above that background editing with the RBFOX2 fusion. We believe that most of these editing from the background editing from the control has to do with error in base calling that's inherent to the nanopore platform that we use as opposed to actual editing events. Because when we look at long reads by PacBio, which has consensus reads with a far fewer error in base calling, we see that these background edits basically disappear, but we maintain this editing around the CLIP regions that we see in the short reads. And with both platforms, we were able to de novo extract the known RBFOX2 motif.
Chris, we have a question from Youssef Barash. How do the motifs you identify for RBFOX relate to the primary and secondary motifs described recently by the Burge lab?
Right. I think we did look at that a bit. It's not in the paper, but I think we picked up at least some of the secondary motifs that were ranked lower for the motifs that we got from the edit clusters. So, we tend to rank the motifs, and the top motif was, of course, the RBFOX2 motif. But then a bit further down, I think we had matching motifs that did rank some of the secondary motifs. But that isn't in the paper.
And we have another question. Let me get to it. Did STAMP versus eClip comparison reveal any artifactual eClip binding? And if so, in what context does STAMP do better than eClip?
So that's a good question. We do have, like I said, edit clusters that do not overlap with clip peaks that are enriched above the background editing that you get from the APOBEC control alone. It remains to be determined whether or not these represent, transient interactions that are bona fide, that clip is not sensitive enough to detect. Where they do overlap with CLIP peaks of the fusion and not the endogenous CLIP, we believe that has to do with artifactual binding from the expression of the fusion as opposed to actual transient interactions. But there are like a handful of, actually a large number of candidate binding sites that could be potentially picked up by STAMP, but that are not sensitive to be picked up by CLIP. At this point, we're trying to evaluate ways that we could verify those binding events are bona fide binding events as opposed to just background editing from localization of the editing enzyme in the region of the RNA. So yeah, there's potential to discover new binding sites that CLIP does not pick up, but we didn't try to evaluate that for this study.
Okay, thanks.
Yep. So for the example gene, Pratibha looked genome-wide at alternative polyadenylation events. She found a large hit list, and I'm just showing you the top hits here for alternative polyadenylation editing by RBFOX2 that you don't see in the control for 2 different isoforms. And I'm showing you here the example gene FAR1. So you have CLIP peak in this FAR1 3' UTR that's enriched above input. For the short reads, you have edits that are enriched above control. And then for the PacBio long reads, you see no background editing in the control, but you have, again, overlapping edits within this region that overlap with CLIP peaks. And then when you split this isoform into 2 different long 3' UTR and short 3' UTR, you see that the edits are basically all segregated to this long 3' UTR, and specifically in this region where the region is shared by both isoforms, you see very specific editing only in this longer 3' UTR. So this is the first time actually being able to view isoform-specific binding events for an RBP, which is pretty exciting.
This is really cool, Chris, the isoform binding. I love it. Have you done any other types of RBPs, maybe ones that could bind in the 5' UTR? Have you seen any footprints of those?
So we did do this with the ribosome subunit that I'll talk about next. This did give us coding sequence edits that were different between different isoforms. We didn't publish it in this paper because we're working on that for a new manuscript. But it is possible to do this at least in coding sequences. There is, for all of these approaches, a slight 3' bias, not only in the edits, but also in the sort of sequencing readouts for both PacBio and Oxford Nanopore. You tend to enrich for reads closer to the 3' end of genes. So detecting things in the 5' end of things might be a little bit more difficult. But we're working on ways to potentially work around that, especially at the single cell level.
Great. It's really cool. Thanks.
Yep. So for the next application, we wanted to fuse to a ribosomal subunit. So-called RiboStamp fuses to a small ribosomal subunit, ApoBec1. And the idea would be that in coding sequences, more highly translated genes would have more edits, more lowly translated genes would have fewer edits. And this is just an example of a highly translated gene. We're showing you a small ribosomal subunit CLIP where you see 5' UTR binding, coding sequence exonic binding, and in the control ApoBec, you see this background editing. Then when you induce RPS2 or RPS3, so these are 2 different small ribosomal subunits, you see that you get a dramatic increase in editing clusters on the 5' UTR coding sequence exons and some signal in the 3' UTR.
Chris, I'm going to stop you again.
Sure.
You have a lot of interest. So Matthew Gazzara is asking, does FAR1 isoform B that lacks RBFOX2 binding make it to the cytoplasm?
We assume that it does. This is a poly-A enrichment protocol for generating these libraries. So we're under the assumption that these mRNAs are fully polyadenylated mature mRNAs that have been exported in a cytoplasm. If there's literature about FAR1 being retained in the nucleus when it has a shorter 3' UTR, I haven't found it.
Okay, and another question from Yusuf Barash. If the areas are shared between the isoforms in the 3' end, why would the RBP only bind that region in one isoform? What kind of mechanism would explain that?
Yes, so we didn't explore the mechanism, but that's a very interesting question. So that could have to do with, isoform-specific features that are recognized by certain partners of RBFOX2 or certain cellular environments of, RBP expression profiles. So there are many possible mechanistic explanations as to why RBFOX2 would prefer one isoform over another. But yeah, that was outside of the scope of this study. We just observed the alternate isoform binding.
Okay.
So just as a proof of concept control, MALAT1 is a non-coding RNA that's highly expressed. It's also cytoplasmic in dividing cells, and we did not see any detectable RiboStamp signal across this non-coding RNA, so it leads us to believe that these coding sequence edits are actually translationally directed. So comparing to standard ribosome profiling approaches, here I'm showing you polysome-seq comparison to the RiboStamp. And so that's just comparing normalized edits across genes, genome-wide, to polysome sequencing readouts. And what you see is, uninduced or controls are very lowly correlated, but you increase this correlation when you induce the RPS2 fusion. So across all these time points, you have good correlation with standard polysome sequencing readouts. So two different readouts, one being editing and one being polysome sequencing, but they're in concordance. And these edits, these RiboStamp edits, are actually responsive to translational perturbations. So here I'm showing you treatment with an mTOR inhibitor, TORN1, which actually globally downregulates translation. And you can see that there's no effect on gene expression. So these are just RPKM counts for TORN treated versus vehicle. But when you look at the edits themselves, you see that TORN treatment actually reduces translation for many genes, or reduces editing by RiboStamp for many genes, which implies it's a translational perturbation directed alteration. And then another way to look at this is just by looking at the different quadrants of genes that are ribosome occupied by polysome-seq. And so this first quadrant would be the most ribosome occupied, second and on down to the fourth quadrant. And we see that there's a very significant reduction in RiboStamp editing in the first quadrant of polysome-seq occupied genes, which actually contains all of the known TORN-sensitive genes. And this is not something that we observe when we do the same treatment in the ApoBec-only control. So what we wanted to do next is evaluate single-cell resolution use for the technology to either multiplex RBPs at low input or to determine binding sites in different cell types. And just to orient you on how this data is processed, basically, we do a 10x 3' experiment, and we split every single cell into its own barcoded BAM file and run Sailor, which is the computational pipeline that quantifies the edits. And in post-processing, we arrive at a final table where every individual cell has its own editing profile. And here we're just looking at the top 200 genes, comparing bulk stamp to single cell stamp for RBFOX2 and controls. And we see good concordance on these genes between bulk and the aggregate of single cell, and then the individual spots or individual cells. And we zoom in on this example gene that has good concordance here, UQCRH. We can compare RBFOX2 clip peak that's enriched above input to bulk RBFOX2 stamp, which has these edit clusters. And when we look at the aggregate of the single cell experiment, we see a nice overlap with these edits. And then what's really cool is we can split this into individual cells for the control and for RBFOX2, and we can see the variable background editing in the controls, and then the individual cell editing events in these individual cells. And just looking at the concordance between clip and the single cell aggregate, we see roughly similar concordance as we saw with the bulk stamp approach. And downsampling down to one individual cell, so just doing a Homer or motif extraction from like 1,000 cells all the way down to one single cell, we can still extract the RBFOX2 motif from one single cell. So it's highly specific even at one cell resolution. So next we wanted to do a multiplexing experiment. So we borrowed from the technology from the 10x platform, this capture-seq. It's basically just a 20 base pair sequence that we insert between the RNA, between the DNA encoding the ApoBec module and the RBP, and it is hybridized to these capture-seq oligos that are actually on the 10x 3' end beads. These are initially set up to capture guide RNAs from gecko experiments, but we designed them to potentially capture the open reading frames of our RBPs of interest. And so if you express these capture-seq containing RBPs 1 and 2, you should be able to mix cells together and then just deconvolute computationally which cell has which RBP. So we did that for RBFOX2 and TIA1, and I'm just showing you UMAP visualization first on the expression landscape where things aren't that clearly resolved, and then using normalized edit score, we see that these different populations of RBP containing cells nicely separate away from the controls.
Chris?
Yep.
What is the maximum number of multiplexing that you've done?
So the answer is 2. We have tried up to 30, and that's when we learned that this capture sequencing approach is not as efficient as necessary for multiplexing up that high. It's not in the paper, but we're working on a new approach to actually increase the capture rate per cell per RBP. And so we're hoping to be able to scale this up to our entire library of RBPs, which is over 1,000.
Great, good luck.
Thanks. So this is a Louvain clustering, just defining these populations as control, background, RBFOX2 or TIA1. And if we look at the top differentially edited genes in these populations, we see that there are these example genes where you see editing not in the controls, but enriched in both RBPs for this example, or editing enriched across these individual cells for this RBP, but not this, and the other way around. So this is a way to look at multiple RBP binding profiles in individual cells across variably edited or bound genes. And then using the same approach to apply to the ribosome profiling or the RiboStamp approach, we did this again with the 10x, same platform, same experiment. And here I'm just showing you RPS2 stamp versus control. And what we did was we identified the population of cells that overlap with the control. We believe these to be lowly expressing or low copy number RPS2 stamp cells. And we can do a filtering step where we basically remove those cells and concentrate on this population by Louvain clustering. And when we look at this population in aggregate and evaluate the genome-wide edit signal compared to polysome-seq again, we see good concordance with standard polysome-seq profiling with the aggregate of our single-cell signal. And then this is just throwing together all these cellular populations, so control, RBFOX2, TIA1, and the ribosome. We see genome-wide beta gene editing profiles that sort of match what we expect, even though this 10x 3' end approach has a heavy 3' bias. We still get this enrichment for coding sequence edits from the RPS2 population when compared to RBFOX2 and TIA1, which are expected to have 3' end binding primarily. And again, just looking at the top differentially edited genes, comparing the ribosome to these two different RBPs, we see that we can evaluate translation in individual cells on these individual genes, and then individual RNA binding profiles in individual cells on these individual genes. And it matches basically roughly what we see globally. So that's sort of the takeaway. We were able to get STAMP to work for translation and for RBP binding sites at the single cell level. And more recently, Jackie Anstein in the lab published the first in vivo use case of this approach. So she did a RBP-centered CRISPR screen in MYC-dependent breast model of breast cancer. And she identified a handful of RBPs that are important for survival of these cells. YTHDF2 is a M6A reader that she probed in more detail. When she depleted this from these breast cancer cells, she was able to reduce the tumor size for xenograft models. And the use of STAMP was to actually evaluate the translational landscape in these YTHDF2 depleted xenograft models. And she saw that depletion actually altered the translational landscape. So this is first in vivo use of looking at single cell resolution translational readout in a breast cancer model. So just to sum up, STAMP can identify binding sites for full-length RBPs. You can do long-read evaluation of isoform-specific targets. You can multiplex at single-cell level, and you can use RiboStamp to evaluate translational landscapes at single-cell. And more recently, we showed that this can actually work in vivo in tumor models of breast cancer. So we anticipate a lot of exciting use cases for this technology moving forward, and we're working on updates. So yeah, cell type specificity in complex organs we expect to be very useful. We expect to be able to use long reads, maybe with different editing enzymes, so A to I combined with C to U to get at cooperativity or antagonism between RBPs and RBP complexes. And then, as I mentioned, for the multiplexing approach, we'd like to scale this up for our entire RBP library. So with that, I'd like to thank again the Yale Lab, which is an amazing place to work, and all the very talented people that did the work that I showed you along the way, my funding, and I'll take any further questions.
Thank you so much, Chris. This was very interesting. I do have a bit of a follow-up question. The in vivo work is really, really cool and exciting. Have you tried doing this on patient-derived samples, or is the transfection stage a bit of an obstacle?
We haven't tried this in patient-derived samples yet. We're planning to do that, so we've constructed some expression constructs that are actually in, AAV delivery systems, which would be more amenable to organoids or maybe patient-derived xenografts. We are, also evaluating the transduction efficiency for the lentiviral constructs that we're updating. So those aren't, as efficient for certain cellular contexts as AAV, but those are in the works.
Yeah, I read that you mentioned the transient transfection of synthetic mRNA. So is that something that you're pursuing right now, or other than that?
Transient transfection of synthetic mRNAs to express the
the fusion, the fusion proteins and then into the cells.
So we haven't tried transfection of synthetic mRNAs expressing the fusions. Things we've tried so far is transient transfection of plasmids expressing, or integration of lentiviral, cassettes into the host genome, or AAV delivery. And we're also toying around with just incubating with recombinant ApoBec fusions. So that would be sort of an in vitro approach that might be amenable to sort of spatial transcriptomics or patient tissues. But yeah, that's in the early stages right now. But transfection of synthetic RNAs is something we haven't tried yet.
Okay, very exciting.
I managed to squeeze it in under time, even though we had that major 20-minute setback.
Yeah.
I guess hopefully I didn't go too blazingly fast for people to appreciate what was going on.
We have another question. I think the speed was okay. Yusef Barash is asking, how quantitative is your estimate of translation levels? How does it compare to RiboSec in that sense?
How quantitative is estimation of translation levels? So the answer is that it is in decent concordance with, RiboSeq. So if a gene tends to have a high ribosome occupancy readout by RiboSeq, it tends to have a high editing readout by RiboStamp. And we can get, perturbation readouts, at least on the time scale of 12 to 72 hours. Quantitation of actual occupancy, so we don't have like codon level resolution that can measure ribosome pausing yet. We're working on potential new ribosomal subunits that might be able to accomplish that. And then translational efficiency, we don't claim to be able to very accurately measure translational efficiency, even though we are able to measure both transcript levels and editing frequency simultaneously. So there could be a computational workaround to actually get at translational efficiency, which is where you sort of divide the ribosome protected fragments or the ribosome edited RNAs by the transcript level and get a readout for efficiency. But we don't have that level of resolution or a codon level of resolution with this current version.
Okay, and we had another question by Matthew Kinzel-Valliere. How much optimization is required for each new RBP tested by STAMP?
It's a good question. I would say that the groundwork that needs to be done at this point, if you have a new RBP that you want to try with STAMP, is first to establish ground truth binding profiles by clip, if that's possible. And then once you've established your fusion edits, then clipping the fusion, and again, localizing the fusion by IF would be recommended to make sure that you're at, roughly physiological binding and localization. And then there's also orientation optimization that can be done. So for Fox, we did try both N-terminal and C-terminal fusions. It seemed to work better for C-terminal. You can also adjust the linker. It's involved. We used 2 different linker versions in the paper. They seem to perform roughly similarly. But yeah, I would say for each use case, I would say that the groundwork, the legwork would be to first establish clip binding profiles if possible, and then you might have to play with expression levels, orientation of the fusion to get the optimal physiological binding profiles.
Great. And we have another question from Xuan Ye. Would there be a limit number for the RBP demultiplexing, since the APOBEC would modify the RNA template? And if there are too many edits, and is there an edit limit for sequence alignment?
So let's see if I understand the question.
You can see it in the chat. It's a
Let
lot of parts to the question.
Okay, would there be a limit for the RBP demultiplexing? So I take that to mean how many RBPs can you multiplex in one single experiment? That would, I think, be limited by how much sequencing you want to do. So in theory, let's just say you could do 1,000. You would just have to sequence, use more 10x kits and sequence more deeply to be able to detect all the different RBPs and all the different cells. But in theory, that's possible. Since each cell gets its own RBP and its own binding profile, you just need to have a read depth that gives you that detection. Since the APOBEC would modify the RNA template, and if there are too many edits, is there an edits limit for sequence alignment? So we haven't had like a saturation issue, I think, of like too many edits to detect or to quantify. I'm not sure that would arise in a 72-hour experiment, which is about as far out as we've gone. I imagine that if you express the fusion for longer periods, maybe up to a week or more, you might just saturate edits across all different transcripts within the cell. So the editing profile might become less specific over time. And I imagine that over time you might get some repercussive, like quality control reactions from the cell if you had many transcripts edited, because you recode them, you target them for NMD, and so there will be some gene expression consequences.